It's indeed a pleasure being here to introduce my colleague and friend, Professor Ruchi Anand. I should perhaps thank our head of the department, Professor Ravikant, for having given me the opportunity to do so. Ruchi has been here with us since 2008. Before that, her education was from Miranda House, followed by IIT Kanpur, followed by Cornell University, after a couple of postdocs in Sloan Kettering and UPenn. She came here, and when she joined us, what we knew about her was that she's a structural biologist, and we knew that she does protein crystallography, which is, everybody said, a very, very difficult thing to do. And then many years have passed since 2008, and Ruchi has developed this outstanding school in which what they do really is that they solve problems of chemical biology using not only protein crystallography, but whatever it takes to do so. So she has been one of the major impactful people who have made it possible for us to get the cryo-EM facility that was inaugurated last week. Other than that, she is into fluorescence spectroscopy. She's done a lot of MD simulation. And the approach, as I understand, is to understand a problem, whatever is required will be done. She has published many good papers, and her work has been recognized in the form of many awards, recognitions, fellowships. She's fellow of all the Indian science academies. She received the CRSI Bronze Medal. She is an associate editor of biochemistry. She has served on the editorial advisory board of Journal of Physical Chemistry, as well as ACS Censors. She is on every possible committee of DST and SARB over the last 5-10 years. She's a member of the Project Advisory Committee of Organic Chemistry, and her presence makes that part of chemistry a little more physical, I suppose. And more often than not, what we see is that we like to build narrow domestic walls around ourselves. We say that I am a physical chemist, an experimentalist, so I'll not think about theory, I'll not think about biology, I'll not think about anything else. Or I'm a biologist, so I'm not going to get into maths at all. So as we understand that our job here is not just research, teaching is a very important aspect. And there, Ruchi has not only developed very unique courses in her core area, but also she has taught core courses in physical chemistry. And I'm sure she doesn't mind if I share that she was actually, she got the best student feedback the first time she taught the 100-level course. So that was mostly on quantum chemistry. So here we have a complete scientist, and it's really nice to see that her work has been recognized in the form of this. I could go on and on with her achievements and all, but I'm sure we'd rather hear Ruchi speak.
All right. First of all, thank you, Anand, for giving such a nice introduction. I didn't even know I was so good. But it felt really good to hear all the things you said. But I would also like to thank the Institute and the committee and my students and my family for supporting me always, my department. Overall, I've got a lot of strong support from many, many people over the years, and most of the things that happens to, or any applause that a person has is all the people are actually responsible. And in particular, before I start even, I want to thank my students, because we are a team, we work together. And it is by collaboration that all this work that I'm going to show today is there. And actually, there is a lot of work I'm not going to show because I wanted to only focus on one topic so that a little bit of snippet of what we have done is there. But there are many other works that we do that I'm unable to share because of the times. I just wanted you to tell what our lab does in a small way and by taking one problem that we are working on today. Okay, so all the students whose work I'm not discussing, I'm sorry. You were there with me, and you are the people who actually make everything happen. So thank you, thanks to them. So here I'm going to tell broadly our laboratory works on two different problems, and our approach is maybe bottom up in the sense that we want to look for new things. There can be people, we have just heard an excellent talk by Rohit, where the translation is happening. Here we are trying to find out what new translation avenues can be actually thought of, right? Because someone has to go from the bottom to figure out what is new that we have to do. So there are all kinds of sects of so that's why I'm the pure sciences category here. So what we do is we try to figure out a problem, and then we try to see if using proteins or enzymes and different kinds of mechanistic studies can we go to the next step and figure out and go towards inching a problem. In that case, we use two problems, one in which we try to figure out if we can develop sensors, which is more applied part of our work, but it stems from a very basic first understanding of how, what's the problem, how to address it. I will not get time to tell you about this. We look at aromatic pollutants which are difficult to detect, to find out strategies for molecules like benzene, toluene, with functional groups in the chemical language. It's not so easy to detect them. So what do you do? Because it's difficult to detect, we use evolutionarily advanced enzymes, and then we work on them to make it better. So I'm not going to be able to talk about that today, but I'll just show you one or two slides. But the second work that I'm going to be talking about is we're going to look at the problem of antibiotic resistance, and that's what my talk will be mostly about. I'm going to tell you about our approach, and ours is not the only approach, but one of the approaches. The question we are trying to understand is why is antibiotic resistance so prevalent, and what are the mechanisms that make normal bugs? You were having, certain bugs were resistant to certain set of antibiotics, and suddenly you start developing resistance to them. So what is happening? Why is this resistance developing? Can we do something about it? Can we look for new drug therapies? Can we design new drug therapies? To do that, first we have to understand why. So we are at the question of why, and if we can understand the why, we can inch towards an answer. And perhaps tomorrow somebody else more entrepreneurial than us can take up that answer and open up a company and add up to the bandwagon, right? So this is the question that we are trying to answer. We are asking why, how, what's going on. So I'm going to tell you one example only of the problem that we are working on today with why do certain bugs become resistant to antibiotics, and what is the possibility how to attack it. So before that, just the aromatic pollutants, we have done a lot of work. We have tried to understand how an aromatic pollutant gets actually detected. We have also worked with making devices with collaboration with chemical engineering and with Deepti Gupta at MEMS, where we make electrochemical sensors for these. So some translation aspects are there. We're now doing synthetic biology-based sensors with Arindam in chemistry. It got stuck. And we are able to develop these very nice looking E. coli where they glow. I shouldn't be saying glow, that's the wrong word. Arindam will kill me here if he hears that. But basically we can have a palette of colors where we can tell one pollutant from other. So here we can do multiple sensing of multiple pollutants in water. So in a single sample you can perhaps figure out if there is multiple aromatic pollutants that are there. So, that work I cannot talk, but let me go back to the problem of antibiotic resistance. Now, if you remember, when I talk about antibiotics, most of you remember erythromycin. I'm not that well today, so not taking antibiotics yet since I'm talking about it, but erythromycin is there, aminoglycosides are there, you're very familiar with these common antibiotics, and they save our lives. The minute an infection cannot go away, that's a big problem. We have a lifespan which is more because we can get rid of these small infections that we have, right? But most of these antibiotics were made by soil bacteria. Soil bacteria, what they used to do is they want the dominion. So they used to come up with a strategy where they used to put out antibiotics into soil so that other bacteria cannot grow. But then some very smart human beings, and many of the names, it's not antibiotics, but Alexander Fleming is known to everyone. Just by looking at the things that are growing, people figured out, wow, these can be used for us too. We can kill the bacteria too. So streptomycin, for example, is from Streptomyces, and 70% of the other drugs that in the market are actually naturally made by these bacteria. These bacteria don't die of their drugs. They actually make them and survive. So that means antibiotic resistance mechanisms are inbuilt. From evolutionary, from the day antibiotics were there were methods to resist them. And that's what the bacteria are using. That's what they gain. And suddenly a bacteria which was so happy and was not happy, we were happy that they were getting killed, gets one of these mechanisms, and then that's the end of the killing. For us, it's very bad, but for them, it's life. So let me tell you the mechanism that I was going to talk about. This is the ribosome. So most of the proteins in our body, in bacteria's body, in every body, are made by these. And this is what is showing is an RNA, it's very fast step, is taking out the protein. Can you see that protein coming out there? Each and every protein comes out like that, whether it's my body, bacteria body, or any body. So then what happens is most of the antibiotics, they stop this protein synthesis from happening. So the bacteria is dying because our protein synthesis is not stopping, but the protein synthesis of the bacteria we are selectively able to stop. Right? So these antibiotics, they go and bind into this machinery which is making the peptide bond. So what is happening here is this is the ribosome again, and these are all the antibiotics which go and bind the ribosome, and we use them on a daily basis for helping with infections. I'm going to talk about only one such class, which is about 50 of them, macrolide, linconomide, streptogramin. And these are the ones that I was talking about. Erythromycin, for example, is one of the 50 drugs which is in this. And what happens in this is bacteria die because in the bacterial tunnel where the protein is coming out, you saw I was showing you the protein is coming out, these antibiotics go and sit there. When they sit there, the protein cannot come out, so the bacteria dies. Right? So what the bugs do is they don't want that antibiotic to go and sit there. Right? If it sits there, then they will die. So they have come up with something very elegant. What they do is, in that tunnel, there is a lot of RNA, and they methylate one of the RNA bases. So basically what is happening is you have a door here, from which you can exit. But if I put a big stool there, then it's always open, right? So something like that. If instead they put a pebble there, so that stool cannot fit. It's just that enough space is not given. So what is happening here is the tunnel has the antibiotic. If the antibiotic is able to sit, then the protein synthesis will stop. But if you have a methyl group, a small pebble there, antibiotic is not able to take the same space. It's like either you sit on the chair or I put a bottle and then you'll say, I'm not going to sit on this chair because there's already a bottle there. Right? It's like a mark. So that's what is happening. And as a result, because of steric clash, what is happening is no longer the antibiotics are working. So this is the mechanism which is kind of causing resistance. What is happening is bacteria is getting these enzymes called erythromycin methyltransferases. There are enzymes which put a methyl group on the base. It's like a small pebble. You can put it, this is a tunnel, and no longer this space is being occupied. So the question that, the problem then is, can we circumvent this? Can we make the bacteria not put this methyl mark? If they don't put the methyl mark, they will not be resistant. And that's what we want. We don't want this reaction, this particular small reaction where the N6 base is methylated to happen. So how do we go about it? There are two strategies, people have been hearing. There are two ways to do it. One way, you take the enzyme, put an inhibitor to that enzyme. What it means is it is not able to catalyze the reaction anymore. Okay? Or there is another way in this case. You stop the production of the enzyme itself. So you do not let the enzyme ever being produced. So what are we trying to do? What is our main aim? Not to let this reaction happen. If this reaction doesn't happen, our tunnel is open and our antibiotic will come and sit. If our antibiotic is able to come and sit there, then the bacteria will die. So we want to prevent this mark. Two ways to do it. I'm repeating it. One, have an inhibitor so that this reaction is not catalyzed, or you have this thing in which you do not allow this enzyme to be made only. If the enzyme is never made, no question of inhibition, right? So these are the two strategies we thought that we were going to use. I'm going to talk about both the strategies briefly. Maybe it's a little too technical, so I'll try to make it a little bit less technical. You can ask me a question if anything you remember at the end of it. But basically, let me tell you the question, what we did. We said that this is a very tiny enzyme. It knows it has to go into that particular space, and do a reaction. How does it know? How does this small enzyme know that I have to go here? It's like I've already made up my mind, that is my chair and I have to be back there, although somebody's sitting there, so there's a steric clash. Shobna. So I'm just joking. So what I meant is the enzyme knows where it has to go, right? So the enzyme exactly knows where it has to go. So how does the enzyme know? So can we figure out the targeting mechanism? So we took two enzymes, and Ruchika is a student who worked on it, where you take a position which is at this point, it also gets methylated, and we took a position which is our pathogenic position I'm talking about, and we said, can we fool this enzyme to go here? Why do we want to do that? Because we want to know how does the enzyme decision-making process happen. If I know what is the decision-making process, I can stop that decision-making process. But if I don't know how the decision is happening, I cannot make it. So we are going back to the basics, where we are trying to say, what's the enzyme thinking? Although I don't know whether it has a brain, but it knows evolutionarily that I have to go here. So I said, take these two photographs, this one and this one. This enzyme goes to the pathogenic site, and this enzyme goes to a very different place. It's involved in biogenesis and making sure ribosome forms. These two enzymes do not cross-react. Can I make this enzyme behave like this enzyme? Then I will be able to know what is the targeting mechanism. So the question I'm trying to do is, how does this enzyme know where to go? So what we did was, we did a series of experiments. And I'm not going to go in detail, but what we did was we tried to figure out the targeting. We took one enzyme and cut and pasted portions of one into another. So basically, the reaction they catalyze is the same, but one goes here, one goes there. So I took some part of this enzyme, put it here, and another one, and said, will you go where I want you to go, right? So that's what we did, and when we do that, what happens is we are able to tell that this enzyme, which would never catalyze this reaction because it was not able to recognize this substrate, which is this adenine molecule over here, can we make it recognize? And that's what we did. We cut and pasted portions of the enzyme, and you can see if you make two such cuts and pastes, take one portion of the enzyme here, one portion of the enzyme here, you will see the methylation is going to the pathogenic position. So again, I'm going to tell you what we were doing. We were taking portions of one enzyme from one part to another, and we were changing the targeting. It's like my mind is being changed. Instead of going and sitting there, I'm supposed to go there. How do I do it? I put in certain markers such that now the targeting happens to that particular place instead of my original place. And so by putting these two loops, I make sure that now this enzyme is actually going to the right place, and you can see by doing this, you started with an enzyme. So here this assay, look at this. What's happening is you have bugs. Once you add this particular methyltransferase, you can see, can you see white things? They are growing. Now you take this enzyme which does not know how to target, they are not growing. But when I cut and paste portions of this enzyme into this, it starts growing. So what? I've given the information of this enzyme into this enzyme. So now I've made a new variant of the enzyme which is also pathogenic. Yeah, that's a bad thing if I've done it, but I have got knowledge. What I've gained out of it, I've got the knowledge how is this becoming pathogenic, which is a very important thing, because now I can design drugs, right? Now I know this is the point of pathogenesis, and if I put a drug right at this site, then the pathogenesis will not happen, because I can inhibit the enzyme. So we are doing logic-based design of drugs. So that's exactly what we tried to do. But before we could do that, what we did was we tried to understand, let's take our logic a little bit more ahead. Let's try to understand that for the reaction, what is very much required is that this enzyme sees the substrate, the ribosome. So what is in the ribosome it's recognizing? I saw what is in the enzyme it's recognizing, those loops I showed you. But what is it? Does the ribosome, it's like what is the shape of the ribosome? Or what is the shape of RNA it likes? Does it like a bent table? Does it like a straight table? If I am coming nearby, what type of architecture? Suppose I will have an architecture preference. So the enzyme has an architectural preference. So I'm going to look at the architectural preference, and we looked at this architectural preference and the mechanism that follows. What we can show here is the architecture is very important. The architecture is not like base. If you remember DNA, everything is base paired. RNA is not like that. Some things are out, some things are in, and these outward protrusions, these things which are out are very important. Architectural preferences, that's what we showed, and you can see if you mess with the architecture, you lose activity. So if you have something which is going like this red thing, very nice synergy between the enzyme and the RNA, and if it goes down, very bad synergy. That means they cannot work together. That means we exactly know by changing this architecture how it's happening. Not only that, we used, like Professor Anand was saying, we used a lot of fluorescence to figure out this architectural preference. And when you have a handshake with an enzyme, it's like the enzyme and the RNA will come, but then some conformational changes, like a handshake. This is my conformational change. So that's what we tried to figure out. And what we did was to figure that conformational change out. We did something called fluorescence experiments, where we can look at the changes in fluorescence, and we can figure out what is happening to the dynamics of the RNA in this case, because we have put the fluorophores on the RNA. And not only that, we can also combine these studies with MD simulation, and in short, when we can try to understand how they're moving in the reaction. Not just that a reaction is happening, but how is the reaction happening. And what this study told us, this is telling you about the dynamics here, but let me see if it all stops in the end. Because of interest of time, I don't know if I'm going to go over. So I'm just going to tell you the bottom line here. You look here and you can see that for any RNA, for a handshake to happen, the handshake happens at the active site, where the base which needs to be actually methylated flips, and another handshake happens 20 angstroms away. So what it is telling you is, it is not like the handshake is only local. The handshake is throughout happening. So if in this case, something has to happen here, something else has to happen over there. And that particular site which is far away is our druggable site now. Why? Because we don't want to put a site right at the active site, because there are many methyl transferases. What we are trying to do here is try to find a druggable site. And that's exactly what we did. And we made sure that druggable site was good, because we did a cryo-EM structure, and we showed again that really two handshakes are required. You need to have a base flip, as they call it here, so that an activity can happen here. So what this whole study is telling is that when you do a catalytic reaction, it is not only the active site, but things very far from the active site also play a very important role. So every time you have to go at the active site, you can go far where there is less interference, and you call it an allosteric site, and you can use that as the site of reaction. So we were able to do a cryo-EM structure. This is a cryo-EM structure of the 30S ribosome so the ribosome is where everything is made, with a sequence, and we are able to show that for most of these reactions of this methylation type, you need the site of action and a very far away site for being able to recognize efficiently this site of action. So if that is the case, that means we have found a druggable site, and that's what we are doing. Damini in our lab is here, and she's doing that. We have been trying to screen a lot of compounds. We have come up with some scaffolds, and not only that, we have been able to show by different rounds of screening that if you now use erythromycin and the drug together, so if it is not working, then the whole bar is up. When you put two drugs together, the cells start dying. So what we are doing is, we are using old antibiotics. So what are we doing so far? The erythromycin is what we want to use. We cannot develop a new drug so easily. But we have found another drug which will just stop the reaction of that methylation. Obviously, we are not there completely. Our numbers are still off, but we have a starting point. We can do with gram-negative and gram-positive. It's easier with gram-positive, obviously, because they don't have a membrane that is so outer layer of glycans. But what it tells us is that if we use our strategy, we can now have a starting point of drug development, from beginning to think about the problem to a point where we can start coming up with using old antibiotics and with known compounds that we have, and we are using a druggable site that we have found. So we are not there. I'm not saying tomorrow I'm going to package this drug to you, because like it was said to me, I need to do all those studies and go on, perhaps improve it further before I do animal studies and go on. But we are at a starting point of thinking about it. So in the last 5 minutes, I hope I have 5 minutes to go. I'll talk about the second problem that we are, the second strategy of the same problem we are looking at. I told you we can inhibit the enzyme. I said we can just get rid of the enzyme expression itself. So in most bacteria, you know, this thing that I was talking about, this methylation, you know, nobody wants to have, you know, if I want a door, I would not like any kind of, if I want it closed, I don't want a pebble or, you know, half open anything until unless it's necessary. I will only want it open or half open if there is, I'm waiting for someone or something is there. So this methylation that I was talking about in that tunnel does not happen if there is no drug pressure. The door closed is better than little bit of door open with a nudge, right? So because that's not what we want when I'm giving a talk. I don't want someone to just slip in after the talk has started, for example, right? So this is what exactly happens. Only when required, these enzymes are produced. When not required, these enzymes are shut. No methylation, you don't want any pebble, you don't want nothing. You just want normal ribosomes to do their job, make nice proteins, catalyze in your body, cut reactions are going to happen. And that's what you want. You don't want a small methylene, each and every peptide thinking, should I be away from it? Should I try to do something? You don't want that. Right? So this is the mechanism in which, very roughly I'll tell you, what happens is when there is no drug pressure, then this ERM-C is controlled by this RNA switch type thing where it is not expressed. So every time an enzyme needs to be expressed, ribosome has to sit on it, and if you do not allow that site open where ribosome has to sit, it never happens. So you can see in general what happens is when there is no drug pressure, ribosome sits on this RBS's ribosome binding site, it sits here, and coolly makes a 19 amino acid peptide, leaves. No pressure. No enzyme is being produced, no methylation is happening. However, whenever there is this erythromycin pressure, you need the methylation, otherwise you're going to die, right, of the drug. So then what happens is this ribosome starts nudging and stops. About 8-9 amino acids, it stops. And it gives time for this hairpin here to rearrange so that this opens up and ERM expression starts. So what this whole mechanism, whether you understand or not, what it is doing is no ERM, right? What do we want? No ERM, right? Because if there's no ERM, there's no methylation. So then what do I want? This never to happen, right? I do not want this thing to come and stop. The minute it stops here, it causes problems for me because it is a signal for expression of this enzyme. If this enzyme is never there, there is no methylation, there is, the bacteria cannot become resistant to drugs, right? So that's what we want. We don't want this methylation to happen. We don't want to give the edge to the bacteria. So what Chris in our lab did was, she said, why don't we make antibiotics which are conjugate. Now we are making new antibiotics slightly, starting with the same scaffold, so that we can not let this particular stage ever happen. So how will we do that? We have this thing, ribosome, and this peptide is coming out. But if we put something already here, I actually stop this process. I actually put some other peptide. Then this peptide is, you know, same space. Same thing as the analogy I gave. If I put something, a laptop on that chair, nobody will sit. So I'm going to just put something on that chair so that nothing, this peptide cannot form. So Chris came up with this strategy along with Swarn Kulkarni's lab. We made many compounds. Actually, this is the parent drug that we all use, clarithromycin. And we came up with peptides with some design. In interest of time, we can always talk later, but we made a library in which we made, what we were trying to do is, we were trying to come up with new drugs which will not allow this stopping to happen and expression is completely killed. That's what we want. So Chris did that. Whether we were able to completely kill or not, maybe not, but we were able to go a step further in which we made many. I will just tell you a little bit of the data. And what we can show here is our new drugs that we made works as good as the clarithromycin. So if you don't have any resistance, you can use this drug and it's working. But we are not making it for that. We are making it for when you have resistance, will they work, right? So this is what the data which Chris has, initial data, where she's showing if you have resistance bacteria already with this kind of resistance, the erythromycin does not work at all. But the conjugate that Chris has made is actually now started to kill cells. Obviously, we are not there. It's not like tomorrow I'm going to put it and package it into the market, but we are already about a couple of 5-6 fold more than that already killing, right? 2,000 to at least below 300 we are there. We need to be much below. We need to be here. So one day maybe we'll reach, but what Chris then went ahead and with the help of cryo-electron microscopy, we were able to show, and this was while we were going for training, so I thank IIT for helping us with it also. While we were training on the instrument that we now have installed, we collected this data, and you can see here, we can clearly see the drug in the ribosome. So we have the ribosome, 50S ribosome, and the thing, and you can see here, I'll show you. This is the ribosome, and can you see the yellow thing? That's our drug, and this is the same drug, the original drug plus our peptide that we have put. So what do we know about the basic mechanism? Because if we don't know the basic mechanism, I don't know how to improve it. And we are going to get there, right, eventually to improve it. So what we did, this structure told us was the very important thing, that if I was having only erythromycin, my leader peptide, which was there, was going, and it was stalling. But I don't want my leader peptide to go anywhere. So my new drug does not allow this to happen. It's sitting right in its path. So once it reaches here, it cannot go there. So, and now again, we have used a steric analogy. If something is already there, the other thing cannot come. And as a result, we can perhaps think from a mechanistic point to be able to now stop this expression of this enzyme, because this whole process that I was talking about, we're stopping. Right? So that's what we were trying to do. We were trying to make sure this peptide that will be forming over there, like I showed you, does not reach its full potential. Right? Before it reaches its full potential, we have our drug come in and just cut it right here, and then use this against resistant bacteria. So yeah, with this, I will stop. And this was not the only project in our lab, but I thought that this is one of the main focuses of our lab that we are currently trying to really nudge towards, apart from the biosensing I talked about. So we were just trying to use logic to be able to come up with strategies where we can use old drugs, erythromycin plus an adjuvant, you can call it in that way. Or we come up with a completely new strategies where we use the old drug as a binding site and kill the resistance pathway itself. So both the strategies are based on trying to be able to understand what's going on, how, why, what. With that, I'm going to stop here, and I'm just going to acknowledge my students again. This is an older picture of our group, but some of the members are right here. And many of them who did the work are now already doing postdocs in different places across the world. But I have many collaborators. We collaborate heavily with IIT Bombay, and actually that is something that I would really think is a plus point, because what we want to create is an ecosystem where we are able to work with people around us. Right? That's what we would really like. We have Pradeep, Kumar P.I., Swarn Kulkarni. We have Naresh, who helped us with and now I'm working with Arindam. We have worked in other projects with Deepti and with Rajdeep, which I have not mentioned because I only mentioned this project, and that's bad actually, since it's supposed to be an overall talk, but I'm saying it now. But we have many people that we collaborate. It's not just IIT Bombay, but we do collaborate outside, but we do try to find the expertise right here if we can, because then free flow of students, free flow of ideas and exchange, and quickness of the process can be maintained, right? So we have many collaborators from IIT Bombay itself, and Vinod at NCBS was very instrumental in helping us learn cryo-EM, because now we have a cryo-EM facility right here, which I'm hoping that a large percentage of people from here and the Mumbai region and the western region will come and use even outside that. We had just recently had a workshop which was very overwhelmingly full. We had to actually stretch it for one day to even allow even IIT Bombay students to be able to get in. But we got people from all over the country for this workshop, where we let them do even break a little bit of stuff in the lab, because I thought it was important that they actually do it hands-on. So with that, I'm sure not only we are going to benefit, like a whole community is going to be helped with the cryo-EM facility. People can come with ideas. And I'd like to really help, we really say Wellcome Trust India Alliance was able to fund this exploratory antibiotic project that we have. And because the 5-year funding, it really helped us. And the STARS project is also a part of that funding. And DST, DBT, Vadwani Research Center, which is not mentioned here, I mean, they will kill me, but for my protein work, IIT Bombay, all of these, all the support that we have got over the years from inside the institute and from agencies have really made a huge difference in our lives, without which we could not have reached where we have. And again, let me thank the students one more time. I hope we can continue to collaborate and work together and get better and better things together. All right, that's about it. Thank you.